A vertebrate with no red blood cells, no hemoglobin, completely transparent blood, and it is alive right now, swimming beneath the Antarctic ice. In 1927, a Norwegian researcher named Ditlef Rustad pulled a pale, ghost-white fish from the Southern Ocean aboard the research vessel Norwegian. The water temperature was below zero. The fish was limp and colorless in his hands, wrong somehow, in a way he couldn't immediately name. It took nearly 3 decades to confirm what Rustad had actually found. In 1954, Johan Ruud at the University of Oslo published a paper in Nature. The finding was blunt. The blood of the icefish is colorless and contains no red blood corpuscles. One sentence. 30 years of confusion resolved. Every vertebrate on Earth carries hemoglobin. Fish, bird, mammal, reptile, every single one. Except this. The creature is Channichthyidae aceratus, the Antarctic blackfin icefish. It lives at depths between 200 and 700 meters in water so cold it would freeze human tissue in minutes. It does not shiver. It does not suffocate. It hunts. It reproduces. It thrives. The Southern Ocean sits near -1.9 degrees Celsius, below the freezing point of standard seawater. Arthur de Vries at the University of Illinois discovered in 1969 that Antarctic fish carry antifreeze glycoproteins in their blood, molecular chains that physically block ice crystals from forming inside tissue. The icefish has those. What it does not have is the one thing every other vertebrate uses to carry oxygen to its cells. No red cells, no pigment, clear as water in a glass. The question biologists asked in 1954 was the same question they were still asking 40 years later. How does this animal not die? The anatomy offered one clue. Its heart is 4 times larger relative to body mass than any comparable red-blooded fish, cardiac output 5 times greater. The body was compensating, but compensating for what, exactly, and how far back the decision was made, remained open. In May 2019, a genome paper published in Nature Ecology and Evolution finally reached back to the source. What the researchers found was not a mutation, it was not a silenced gene, or a degraded sequence. The answer was something no adaptation story had prepared them for, and that answer is in the genome itself, confirmed, permanent, and unlike anything found in any other vertebrate on record. Here is the obvious explanation. Cold water holds more dissolved oxygen than warm water. The Southern Ocean, near freezing year-round, is among the most oxygen-saturated environments on Earth. So the logic writes itself. If oxygen is already abundant in the water, maybe hemoglobin becomes redundant, a costly protein metabolically expensive to build. Cut it, and you streamline. It is a clean argument, elegant, even, and for decades, it was the working assumption. Dissolved oxygen in Antarctic surface water runs 8 to 9 milliliters per liter, measurably higher than tropical seas. A researcher running a meter through near-freezing seawater in the 1980s would watch the reading climb and feel the theory confirm itself. The ocean was doing the work. The fish just had to breathe it in. But the math does not close. Even in oxygen-saturated Antarctic water, dissolved oxygen alone can only carry about 3% of what hemoglobin-bound oxygen carries in a normal vertebrate. 3%. Icefish have no hemoglobin and no red blood cells, and yet they sustain full vertebrate metabolism. The gap is not small, it is not a rounding error. H. William Detrick III at Northeastern University spent years mapping how icefish actually survive. The answer was not efficient simplicity, it was compensatory extravagance. Hearts 4 times larger relative to body mass than related red-blooded fish. Cardiac output 5 times greater, blood vessels significantly wider. The icefish did not reduce its energy demands, it rebuilt its entire circulatory system from scratch to compensate for the absence. That is not a workaround, that is a catastrophe. Survived. Then came May 2019. Chi-Hing Christina Cheng at the University of Illinois led a team that sequenced the complete genome of Channichthyidae aceratus. They were looking for the hemoglobin genes, where they had degraded, how silenced, how dormant. The genes were not degraded, they were not silenced. What Cheng's team found in that genome was something no one had expected. and it did not fit the word adaptation at all. Here is what the 2019 genome paper actually found. Not a mutation, not a silenced gene, not a degraded copy sitting dormant somewhere in the sequence. Chi-Hing Christina Cheng at the University of Illinois led the team that mapped the complete genome of Channichthyidae aceratus, and the hemoglobin gene loci were absent entirely, gone from the chromosome as if they had never been written. That is not how evolution normally works. Genes get corrupted, they get switched off, they accumulate errors over millions of years until they stop functioning, but they leave a trace, a remnant, a ghost sequence. The icefish genome contained none. As Cocker and colleagues documented in 1,995 inches PNAS, the loss of hemoglobin genes represents an evolutionary event that is essentially irreversible. No recoverable copy exists anywhere in the lineage, across all 16 icefish species. Every single one descended from a single ancestor that crossed this threshold 15 to 20 million years ago, and the door closed behind it. The deletion did not kill the lineage, it rebuilt it. H. William Detrick III at Northeastern University documented what the body constructed in hemoglobin's absence. A heart 4 times larger relative to body mass than any related red-blooded fish. Cardiac output 5 times greater. Blood volume expanded to compensate. Capillaries widened until oxygen could diffuse directly through vessel walls without a carrier molecule. The icefish did not adapt around the loss. It engineered a completely new cardiovascular architecture because of it. The deletion was the blueprint. And the antifreeze glycoproteins Arthur de Vries first characterized in 1969 remained, circulating through transparent blood, binding ice crystals at the molecular level before they could form. A system built entirely without red cells, without hemoglobin, without anything a vertebrate is supposed to require. One vertebrate lineage, one irreversible deletion, one completely novel body plan assembled from the wreckage. What the index concludes: the icefish genome is not a record of what was lost, it is a record of what an organism can become when the loss is total, permanent, and 15 million years old.